So I'm going to give you a little bit of background about cancer genetics in general, and then tell you more about the methodology that we use to study cancer genetics, and then how we applied some of these methodologies to PMP and appendix cancer. So for those of you who are not geneticists and are not scientists, I always like to present this slide when how a normal cell can become malignant, at least from the genetic perspective, and it is mostly due to environmental factor or genetic factor or injuries that delete some genes or modify some genes in your normal cells. And those genes can be categorized into two categories. Some oncogenes, those mutations will activate them, and by activating them, they will enhance the proliferation of the cells that will become malignant. But you have another category of genes that are tumor suppressor genes where an alteration, a genetic alteration, is like pretty much removing the brakes. So these like little two logos are from the brakes and the gas pedal of your car, right? So if your brakes are not working, you're going to keep proliferating, you're going to keep moving. And the same thing, if your gas pedal is stuck, you're going to keep moving. Okay, so that's a great metaphor for you to understand oncogene and tumor suppressor genes. Well, of course, those genetic alterations, they don't happen all at once, right? And there is a progression that can be summarized here, where a normal cell will acquire one mutation and another mutation and a third mutation, and then at the end, there will be sufficient mutation that an oncogene will be hit or a tumor suppressor will be hit, and then the cell will become malignant. So, and this pattern actually has been proposed in '92 by Dr. Vogelstein from Johns Hopkins. In colorectal cancer, for example, he described how the normal epithelial can become a full-blown colorectal cancer through successive genetic alteration in many genes. APC is a tumor suppressor gene, KRAS is an oncogene, P53 is a tumor suppressor gene. So he needed this sequential alteration to become full-blown cancer. So what type of alteration are we talking about here? So here is a chromosome that you see on the top. And here are some genes on this chromosome, gene A, B, C, or D. You can actually have a single point mutation in gene B, and that will be sufficient to convert gene B into an oncogene or into a tumor suppressor gene. You could have an insertion, which is a little piece of DNA that's a little longer. It's not a single point mutation. You can have a deletion. And then you can have like bigger scale type of alteration where, for example, you introduce a new gene or a gene that comes from another chromosome and becomes here between A and B. Or you remove entirely gene B, for example. Or you can have gene B and C that becomes inverted. It can be quite messy what's happening in a cell, especially as it develops in a cancer. Okay? So this is quite complicated to study, but fortunately we have some methods that I'm going to talk about. But how do we now classify all these oncogenes and tumor suppressors, and how do you understand how they contribute to the cancer and to the development of the cancer? And in fact, Dr. Bob Weinberg has classified what we know about these genes and how they are sorted in the cell. Some genes, for example, are used to by the cell to trigger cell death. When a cell is sick, they want to go in apoptosis. And some cancer genes, some oncogenes and tumor suppressor genes are actually perturbing this process so that the cell cannot die anymore, so it keeps proliferating even with those aberrancies. Some other pathway, what we call pathway, for example, has to do with the signaling. Some cells like to receive signals that there is food in the environment to keep proliferating. And some cancer cells have found a way to enhance this signal, to amplify the signal, and to take advantage of all this signaling that's happening. So these are what we call the cancer hallmark, how we classify a little bit the pathway that drive cancer, and where those genes are altered and which pathway they're altering. And in fact, this is the secret sauce to what you call personalized medicine and precision medicine. The more we understand about the cancer, the more we can design drugs to actually perturb the alteration or restore the normal function of these different functions. And when you look at some recent development in personal targeted therapy and personalized medicine, you will see that we have drugs now that will inhibit these signaling, for example, to calm down those cancer cells and to eventually kill them. Or, for example, to restore the apoptotic potential of the cell. So This is why we're really interested in learning more about the genetics and the molecular alteration of these cancers, so that we can now propose more personalized, more precision medicine approaches to treat these cancers. Okay, so I'm going to a little bit overlap with what Dr. Valasek has just talked to you, but in a very simplistic way, probably, because this is what I needed to understand in order to help Andy and to move our research project. I needed to understand what this disease was all about and why we were interested in the genetics. So, of course, you're going to hear from a physician today about more the clinical part of it, and you've already heard everything about the pathology. But from what I understand, so first, it's a very hard disease to study. Why is it, especially for genetics? Why is it hard? Because it's rare. About 1 in a million, right, in the US. So we're fortunate enough here to have Andy as a catalyzer and recruiting a lot of patients here. So we have enough samples to do some studies. It's very low cellularity. You saw some of these slides where there are very few cells, and they're very diluted into many different normal cells or stromal cells. They're highly mucinous, so they're really sticky. They're difficult to extract cells, to extract DNA from these samples. And finally, there's no cell line. There's no mouse model that we can use to figure out what treatment is going to work, et cetera. So it makes it really, really hard to study, at least from the molecular standpoint. The treatment, you're going to hear more about it today, but it's really the cytoreduction, the chemotherapy, the HIPEC chemotherapy, and if it recurs, I think sometimes because it's so close to a colorectal cancer, we just use some of the protocols that are used in colorectal cancer. So here is a picture of what the PMP can look like with all those mucinous deposit that invade the peritoneal cavity, but it really all started here at the level of the appendix when it bursted open, and then these are these samples that really we have access to perform our research, okay? So what were the questions that we had when we started to wonder about the genetics of this PMP? It was really, how similar are these alterations to colorectal cancer? After all, the appendix is a little piece of your intestine, right? Are there differences at the molecular level between the low grade and the high grade, like Dr. Valasek just mentioned? And finally, can we understand which pathway are altered so that we can come up with maybe new treatment options, more personalized treatment options? So this is really what was the goal of it. So here is a very, very simplistic one-slide summary of the previous talk, and with a lot of omissions, but at least that was sufficient for me to understand. So we're all talking about mucinous neoplasm of the appendix, which is this disease. It's a subtype of appendix cancer, if you remember. There is the primary, which is really the appendix lesion itself. It can be high-grade, mucinous adenocarcinoma, or it could be this lamine that Dr. Valasek mentioned, low-grade primary, really. But then what we really have access to, especially here at UCSD in a regional reference center, are those lesions after they've been diagnosed as a primary. We get them when they reach the stage of PMP, pseudomyxoma peritonei, which is the syndrome. But in fact, the sample that we get is called mucinous carcinomatosis peritonei, the MCP. And those MCPs could be also low-grade or high-grade. Okay, everybody? All right. Next slide. Oh, yeah. So this is, again, I overlap a little bit with the previous talk, but there is this low-grade MCP where you see this strip of neoplastic mucinous epithelium. And the pathologist will describe that as a low-grade cytologic with some architectural features and abundant extracellular mucin. And this was an example of a high-grade PMP where you see that the cancer cells are more structured. They form those aggregate floating neoplastic mucinous epithelium. They have a more complex architecture and high-grade cytologic features with like bigger nuclei, etc. So that's what we had to start from with as a sample. And so now what are the techniques that we can use to get to the DNA of those samples? Well, first, because they're so low cellularity, we really need to dissect them. So we perform what's called laser capture microdissection, where we really cut out the cells that are really interesting to us. And then we can look at them one gene at a time, okay, by performing what's called a PCR amplification, and then we can read this assay. The problem when we start doing that is that we need to have very strong assumption. We need to figure out, okay, we're going to look at this gene. Well, if it's negative, then it doesn't advance the project. So it's very low throughput, and it needs this dissection, which can be cumbersome and can take a long time, especially if you need a lot of DNA, okay? There's another method that's maybe now more modern, where if you don't want to do the dissection, you can throw the entire sample into your PCR amplification, but then you're going to sequence it using one of those machines that Illumina is making, these very high-throughput, deep sequencing. In fact, you're going to sequence the DNA so many times that even if your mutation is present in 1% of the cells, you're still going to see it as part of these results. So this is kind of the sequence of DNA here, and you can see that this G, you can't see it probably, but this G is turned into an A in about 2% of the cells, and we can clearly see the signal here, okay? So that will avoid us from using this dissection. So that is pretty useful in studying this disease. There's another method that we use that it doesn't require the sequencing, but requires now a different type of PCR, one for the mutant version of the DNA and one for the wild-type version of the DNA. So the same thing here. We perform a PCR, an amplification of the gene, but we have different type of reporters. For example, the mutant gene will give us a red product, and the wild-type gene will give us a green product. And then because we perform all these reactions in mini droplets, like in an emulsion, we can sort all these mini droplets through a machine, and we can plot the red fluorescence versus the green fluorescence. The one that do not shy are droplets that are empty, that didn't work, but we can measure the fraction of normal cells to mutated cells, from wild-type DNA to mutated DNA. And this method is very sensitive. Again, if you only have 2% of your DNA that's mutated, you will be able to see it using a method like that without requiring the microdissection. But again, this method is very low throughput. It only allows you to interrogate one gene at a time. It's actually even worse. It's one mutation at a time. So you already need to know where to look, okay? So the final method that we did, and that's what we did in our project, it's a high throughput method. We wanted to look at the entire genome, all the genes in the genome. And so this is why we still had to use the dissection, so to enrich for the tumor cell, but we perform a method that's called exome capture, where pretty much, so your genome is about 3 billion base pair. We only looked at 1% of them, but that's the 1% that matters. That's the 1% that codes for genes, so tumor suppressor and oncogenes, okay? And so we are fishing out 1% of the genome. We're using this method, and we're throwing this on this deep sequencer that Illumina makes. So what was the result of this project? We looked at 10 different samples of PMP, 1 high grade, 9 low grades, and we found about 25 genes. We found 25 genes that were mutated in at least 2 samples. So that's good because the recurrence for us, if it's mutated in more than 1 sample, that tells us, well, there's something about this gene that's special, okay? In fact, the top 2 genes here were mutated in 9 or 10 out of 10 samples. So it's really significant. It is really a signature now of this disease, okay? And some of those genes are known to be involved in cancer. They are not random genes. Actually, the top gene is one of the most famous oncogene in colorectal cancer, for example, and in lung cancer. And we also noticed a few differences with colorectal cancer. For example, we know that colorectal cancer, which is usually more higher grade, always has a p53 mutation or APC mutation, these 2 tumor suppressors. This appendix cancer, of course, we had only low grade or mostly low grade. They don't have that. And GNAS is absolutely not really seen in colorectal cancer. Okay, so this is the difference with a few other genes are known to be involved in colorectal cancer as well. So this was reassuring to us that there is something real about it. We expanded this analysis. Now we know where to look at. We have KRAS, these top genes, and the GNAS, the number 2 gene, okay? So we know where to look at. So we could use some of these other methodologies to go back to those genes and interrogate more samples. In total, we interrogated 29 samples. And we interrogated 23 low grades and 6 high grades. And we could find differences. Well, first, low grades and high grade, they all have KRAS. The vast majority, more than 80% of the patients have the KRAS mutation in the low grade or high grade. But when you started to compare for GNAS mutations between low grade and high grade, we found that mostly the low grade had the GNAS mutation, and only 1 out of the 6 high grade had the GNAS mutation. So that was interesting to us. We found some difference. And this difference, at least the way we see it, and in the samples we studied, what it means is probably that the high grade, at least in the cases we studied, the high grade did not come from the low grade. Because if they came from the low grade, they would have the GNAS mutation, okay? The fact that they don't have the GNAS mutation means they probably arose independently. They did not progress. So at the time, so we published this result in 2014, about a year ago now. And at the time, there was not much known about the genetics of appendix cancer of PMP. And even now, there are not that many studies. First, because access to samples is very difficult. There was a study done, so these are the different studies, and this is our study here, okay? So I can show you the different things that I tried to compare. So the total number of samples, we had 29 samples in our study, and we found the vast majority of the lamin being mutated for GNAS, but only 1 out of 6 of the appendix cancer mutated for GNAS, and finally the vast majority of both lamin and full-blown cancer, or high-grade, being mutated for KRAS. But one particularity, again, is that we were looking mostly at the PMP, so the peritoneal part of the disease. We did not have access to the primary lesion. These other studies, they did not perform a genome-wide. They did not look at all 20,000 genes. They looked at only KRAS and GNAS, most of the cases, okay? This study here, published at the beginning of the summer, looked at a little more, GNAS, KRAS, BRAF, beta-catenin, and the third promoter. And there's discrepancies, okay? We don't all agree. We don't all agree. We all found some level of mutation of GNAS and KRAS, but really we are at the top here. Some of these other studies only found it maybe in 50% or maybe 70% of the samples, okay? One difference could be the methodology. Maybe they did not do the dissection. If you don't do the dissection, it becomes very hard. You need to be very good to pick up those mutations. So maybe they are false negative here. Some of these other differences is the type of samples they looked at. So we look mostly at PMPs. They look mostly at primary lesion, at least in some of those studies. So who knows? Maybe that can make a difference. And finally, you know, I classified really lamin and high grade, but as you saw from the previous talk, it's a little more complicated than that. There could be some subtleties, and we didn't took advantage of that. But these binary mutation, like both KRAS and GNAS, really the main two molecular features of this particular cancer, is not uncommon. It's quite uncommon, but it has been seen before in other cancer. In fact, it's been seen in a pancreatic cancer that's also, or a pancreatic neoplasm, let's say, that's also highly mucinous, IPMN. It's also secreting a lot of mucin. And in these cancers, there are more than 70% of the patients that have both KRAS, which is very frequent in pancreatic cancer, but KRAS and GNAS, okay? So that's interesting. Usually, pancreatic cancer only has KRAS. In a pre-malignant lesion in adenomas, we see that the villus adenoma also seems to look a little bit like this, with both KRAS and GNAS mutation. And we know that the villus adenomas, in fact, they secrete a lot of mucin. So there seems to be something about having KRAS and GNAS and being like a mucinous cancer. And in fact, those cancers, they grow relatively slow. And that could be also a characteristic of this particular molecular landscape, or that's given by this particular molecular landscape. And so I'm not going to go beyond this level of complexity, but just to explain you how complex it is to make sense of this molecular landscape and this genetic landscape. In fact, KRAS is at the top of this signaling cascade that has been maybe the most studied in cancer, okay, the MAP kinase cascade. So we know that KRAS is an oncogene that's kind of downstream of those signaling receptor. And it activates this cascade that pretty much tells the cell to proliferate, okay? And downstream of this cascade, there's some other enzyme like MEK or ERK, and that eventually triggers the signaling in the nucleus and the program to proliferate. GNAS, interestingly, has also been studied, but not necessarily in the context of cancer. GNAS is part of a cascade that's called the cyclic AMP PKA cascade, and that's involved in secretion. And exocytosis, the PKA enzyme involved in secretion and exocytosis. So no wonder that these cancers that activate GNAS now secrete a lot of mucin, for example. And in fact, as a side note, GNAS and this particular PKA pathway is one of the targets of the cholera toxin. Cholera toxin makes you secrete a lot of water in your intestines, I guess, and that's what is triggered by the toxin. So it's also used in other diseases. Well, so this is not as simple, because we know that there's a lot of interaction between those two pathways, a lot of crosstalk, okay? So if it was that simple, we could maybe find a good drug for it, but we know that this pathway kind of repress this pathway, but then this pathway represses this pathway well. So they kind of cancel each other a little bit. So that explains why maybe some of these cancers are slow growing and slow proliferating. And finally, in some of the cases of these PMP that we analyzed, like in 1 or 2, we also found evidence that other cancer pathways, cancer signaling, are activated. The Wnt signaling, the Hedgehog signaling, the TGF-beta signaling. And those are also good pathways in colorectal cancer. And they contribute to bringing normal cells into cancer cells. And those two, the RAS and PKA pathway, they both contribute and perturb also this signaling in a kind of a contradictory fashion. So it's both applying the brake and the gas pedal at the same time. So it's really much a hairball, and it takes a lot of work and a lot of a cellular model, a mouse model, to really decipher what is important for this particular cancer. And that's some of the studies that we probably want to do. So the final slide here is really about, so what does it do for you guys and for the patients? Well, you're going to hear from Dr. Fanta about what he does with Foundation Medicine. Foundation Medicine is one of those companies that sequence the DNA of those tumors and tell us about which genes are mutated, okay? And we've sent some of the cases to Foundation Medicine, and of course, very frequently they come back with GNAS. And what Foundation suggests, and that's also what we suggested, is that because of this interaction between the RAS and the GNAS pathway, they suggest some of these clinical trials that are called MEK inhibitor clinical trials. If you remember, MEK was one of these enzymes that's downstream of KRAS here. So if you block here, you might be able to cut down the signaling and cut down the proliferation. Okay, so that's what they suggest. There's no result yet. I don't know how well it works, but at least there's a rationale for it. Okay, there's a molecular rationale for it. And you will notice that those trials are actually not open for appendix cancer specifically, because I don't think any company wants to open a trial for appendix cancer. You just too few of those, but they're what's called umbrella trial, where they include many tumors, maybe solid malignancies or metastatic cancer. So umbrella trial. All right, so take-home message. The mucinous appendix cancer has a different genetic landscape than colorectal cancer. The majority are KRAS mutated. In colorectal cancer, it's not the case, about 40% only. So that means that some of the drugs that we use in colorectal cancer, like a GFR inhibitor, for example, are not appropriate, because we know that people who carry this KRAS mutation will not respond to the EGFR inhibitor, for example. We think that, at least in our set of samples, we have evidence to show that the high grade might not progress from a low grade. They might occur independently. But really, the main two results of this study is that both oncogene KRAS and GNAS are mutated. Our interpretation is that it fits with the fact that this cancer secretes a lot of mucin. It fits with the fact that this cancer is slow progression with little invasion. And it's a good candidate for those MEK inhibitors. And finally, we also saw that, like in colorectal cancer, some other signaling pathways are affected in some more rarely, but they are. So the Wnt and TGF-beta and the Sonit Hedgehog pathway. And finally, I would like to thank all the collaborators that contributed to this study. Katie Fraser, Dr. Alakus is really behind it. He's a surgeon from University of Cologne who was performing a postdoc with Dr. Louis and with Dr. Fraser, and really pushed this particular project. The entire lab of Dr. Louis for providing samples and helping us with some of the analysis. And finally, the PMP Research Foundation, which really supported some of this work. and the Cancer Center itself and some of the money from the fundraisers we have that really went into this work, because it's very hard to find funding for this type of work because of the rarity of the disease. So any funding helps. All right. Thank you.
Hi. Work is fascinating, absolutely. It sounded like you started with 10 samples, then an additional 30 after you got your signals. How much does it cost to run one sample approximately?
So no, we started with 10 samples and we added another 19. So that was 29 total. How much does it cost? Whole exome sequencing can cost about, so we're talking about research here. We're not talking about what Foundation Medicine bills to insurance, okay? Because this is different standards. But in a research setting, performing a whole exome can cost anywhere between $1,000 and $2,000, plus analysis, you know. And the analysis is sometimes, you know, maybe 6 months or a year of a postdoc or of a programmer and my supervision. And so, yeah, so these projects, I don't know, it was probably like a, I don't know, if you had to put a number behind it, 50,000, 100,000, something like that. Close to $100,000 for this project, yeah. Yeah, if you start summing everything up.
How much does changing technology interfere, change with your own studies? And technology is changing so fast, it gets in your way, it helps you, it confuses you or whatever. Is that very much a part of your studies? Is evolving technology is part of, aside from your own studies?
I develop technology, I develop new assays, and it really opens gates, right? I mean, these are studies that we couldn't think about doing before, really. And so as soon as we were able to sequence an exome, then we start thinking about, we dare to ask questions that we didn't dare to ask before.
How recent is that? You just said no.
I'm sorry?
How recent was that you just...
How recent?
That you were able to do this?
5 years old, 6 years old.
Thank you. I have a question about how well are you interfacing with all the other research centers on the research? Because I just wonder how coordinated is the overall effort? Is anybody duplicating effort here?
Could be. A lot of the other groups are actually in Japan, so that becomes a little more complicated to coordinate, you know, internationally. I believe that some other centers in the US are interested in that. Actually, as soon as this was published, I presented the result at the American Association for Cancer Research, and a lot of investigators have samples in hand, but don't necessarily have the skill set to do it. So they are interested in collaborating. They're interested in sharing their samples. I was approached by someone from MD Anderson, for example, or from Stanford, who have access to these samples. So, but I believe there is duplicated effort. I think that it's possible that Dr. Sugarbaker has had some project like that. I don't know. It's hard, you know, between the time you think about, oh, yeah, let's do that, and actually being able to do it. It's probably better if several people try. Competition is a good incentive.